Okay, so this video is about phage therapy. And phage therapy dates back to the 1800s, 1900s, where both French and English scientists discovered this therapy. In 1896, Ernest Hankin, a British bacteriologist, reported an antibacterial activity against Vibrio cholera. He suggested that an unidentified substance limited the spread of the cholera epidemic. 2 years later, in 1898, a Russian bacteriologist named Gamaleya observed a similar phenomenon while working with Bacillus subtilis. Through their work, these scientists discovered that phages had the potential to kill the bacteria that causes many infectious diseases in humans, in animals, and even in plants. Throughout the years, several other scientists made similar observations, but it wasn't until 1914 that Frederick Thwart, a British bacteriologist, advanced their hypothesis to the possibility of viruses. But with limitations in finances, Thwart couldn't test his hypothesis. A Canadian microbiologist, Felix de Herel, was the first one to observe the bacteriophage in 1910. He discovered it while studying methods of controlling locusts in Mexico. He went to his lab and spread cultures and observed round zones that had no growth. He called these zones plaques and stated that they were caused by viral parasites. 6 years later, he coined the term bacteriophage or bacterium eater. In 1917, de Herel began testing his phage in human patients. He was under the supervision of Professor Victor Henry Coutinel in Paris, and he demonstrated his phages by ingesting them. Days later, he demonstrated them in a 12-year-old boy who had severe dysentery. His symptoms ceased after a single treatment and made a complete recovery. Through this, de Herel had 3 additional patients suffering from dysentery. He gave the anti-dysentery to them, and within 24 hours of treatment, they began to recover. The first actual reported phage-based clinical trial occurred in France in 1921 by Richard Bernoulli and Joseph Mason. They used bacteriophages to treat staphylococcal skin disease. They treated them through injecting the phages into and around surgical opened lesions. The infections within their patients regressed within 24 to 48 hours. So then over the next 10 years, thousands of people were treated with various phages for other infections, including cholera and even bubonic plague in India. Several hundred studies of phage therapy followed, but it was mostly in Eastern Europe and the former Soviet Union. In 1930, there were 12 available phage therapies. 5 of them were produced through de Herel, and 7 were produced in the United States through the Eli Lilly company. However, these phage preparations were very controversial, and clinical studies were not pursued in the United States or Western Europe. And by 1940, penicillin was introduced, and there was little to no phage therapy studies being conducted. Recent years, interest in phage therapy has grown. This is largely due to the fact that we have an increasing problem of bacterial antibiotic resistance. Phage therapy and antibiotics are similar, but they're very different. Both phage therapy and antibiotics have target ranges, but the difference is phage therapy is very specific, and the chances of developing secondary infections are very low and often avoided. With antibiotics, the target is both pathogenic microorganisms and normal flora in the body, which often leads to secondary infections. The replicate at site of infection and are available where they're most needed, and antibiotics are metabolized in the body, they're eliminated from the body, and therefore don't necessarily concentrate at the site of infection like phage therapy does. To this date, there has been no reported serious side effects of phage therapy. Unlike antibiotics, they have multiple side effects, including intestinal disorders, allergies, and secondary infections like yeast infections. Phage-resistant bacteria remain susceptible to other phages within a similar target range, and resistance to antibiotics is often wide range. Multiple antibiotics with similar phages or with similar mechanisms of action will become ineffective once a resistance develops. The greatest advantage to phage therapy is that new phages can be produced within days or weeks, versus antibiotics, it takes up to 2 decades to get an antibiotic out there. Phage has continued to be used in Eastern Europe and in the former Soviet Union. The pharmacokinetics of therapeutic phage preparations is not well understood still, which is why it's considered experimental in the US. How it works is after a single dose, the phage gets into the bloodstream, and within 2 hours or 4 hours, within 2 to 4 hours, the phage gets into the bloodstream, and then within 10 hours, it gets into the internal organs. The phages insert their viral DNA into the bacterial host cell to produce progeny phages, and then the progeny burst from the host cell, thereby killing the cell. The young phages then proceed to infect more bacteria, and phages can remain into the bodies up to several days. Once the bacterial infection is eradicated, the phage dies, and it leaves no residual virus in the human host. Phages are known to be species specific, and they only infect one or a few species of bacteria. They can be isolated from nature and can be potentially developed in laboratories against any bacterial infections. They've discovered that phages can't parasitize other viruses. They have been administered by the following routes: orally through tablets or liquid forms, rectally, locally, even in tampons, rinses, and topical creams. They've been used in aerosols and intrapleural injections and also through IVs. So far, phage resistance has not been viewed as a major problem because it's so much easier to develop a new phage than a new antibiotic. In nature, bacteria evolve resistance, but then their phage parasites will also co-evolve and adapt. They're saying that this behavior can be duplicated in the laboratory within a few weeks. So the development is much faster than antibiotics, which is why this phage therapy is being pushed. Dr. Blair can use this video as he sees fit, and all of this information can be found at bterfoundation.org.